This is Peter Karp from SRI's Bioinformatics Research Group, and today I'll be talking about a new tool for analysis of metabolomics data called the Omics dashboard. An overview of this tool is that it's an interactive visual tool for analysis not just of metabolomics data, but also of gene expression data and proteomics data. And it has several use cases. One is to quickly survey how all the cellular systems are responding to a given stimulus at multiple levels of detail, from the level of a specific pathway to the area of a larger subsystem of the cell. And it's also a way of, another use case is to gauge the relative levels of different metabolites in the cell. And this tool is now available at biopsych.org and also is available as part of our downloadable Pathway Tools software. I'm going to start out with an overview of both biopsych and Pathway Tools. The Pathway Tools overview will come a little later. Biopsych is a collection of now 11,000 different pathway genome databases for different sequenced organisms. It's mostly microbial, and it consists of predicted metabolic reactions, pathways, and metabolomes, as well as atom mapping information and a few metabolic models. And the information in biopsych has been curated from 66,000 publications. Biopsych has extensive bioinformatics tools for metabolomics analysis, including the ability to paint metabolomics data onto individual pathways, onto multi-pathway diagrams that we call pathway collages, and onto full metabolic map diagrams. And I'll show you that latter capability at the end. We also have a tool within biopsych called Smart Tables that allows you to manipulate metabolite sets in various ways, like very easily mapping a set of metabolites to the corresponding set of metabolic pathways. And these tools are all described in this publication in the Metabolites Journal from 2015. But let's now proceed to talk about the Omics dashboard. Let me start out by talking about the organization of this tool. The dashboard consists of a set of panels and a set of plots. So here we see 4 different panels. There's a panel for biosynthesis, for degradation, energy metabolism, and other metabolic pathways. And within each panel are a series of plots. Within the degradation panel, there's a plot for amino acid degradation, for nucleotide degradation, amine degradation, and so on. The data that we're looking at here are a time series data set for Helicobacter pylori after exposure to metronidazole, an antimicrobial compound. And the values, the metabolite abundance values, are fold change values relative to time 0. And in each plot, each vertical line is showing information from one time point, and each point on a vertical line is one reading for one metabolite. And the set of metabolites on one vertical line are all the metabolites in that subsystem for which data is available. So you can't really, at this level, resolve these visual points too well, but the idea is that we're plotting here all of the metabolites involved in amino acid biosynthetic pathways within each of these vertical bars. And finally, the large point is the average of all the metabolite values shown for that plot. So by letting your eye track the average, it's relatively easy to look at the overall response of these different systems to see, oh, amino acid biosynthesis is increasing and then dips at the end. Amino acid degradation also increases over time, whereas the pentose phosphate pathway is decreasing over time. Now, what happens if we click on the amino acid degradation plot right here? Well, we'll get a new display, which is a new panel that shows now the metabolite levels in all the amino acid degradation pathways in the cell. So we see here alanine degradation, arginine degradation, and serine degradation at the end. And as before, each dot shown in the plots is a metabolite level. So there are metabolite levels for 2 different metabolites available for alanine biosynthesis. And again, the large dots are the averages. Now, if we click on arginine degradation, we'll get this display here, which shows now the abundance levels for all the metabolites in this pathway over time. And those the metabolites that are, for which we have data in this pathway are proline, arginine, urea, and ornithine. So we can see that proline is decreasing significantly, whereas arginine is increasing significantly over time. And finally, if we click the Show Pathways button here, we'll get a pathway diagram that shows us that actually there are 2 arginine degradation pathways in H. pylori, and it shows us next to each metabolite the expression levels, abundance levels of the metabolite over time, with the levels mapped to the color scale as shown over here. All right, now the set of panels and plots that are available in the Omics dashboard are actually configured automatically, both as a function of the current organism and as a function of the current data set. So for example, if we were looking at a photosynthetic microorganism, it would have a photosynthesis pathway available as a plot that would not be available for other organisms. On the other hand, if there were absolutely no data for that particular pathway, then in a given data set, then the display of that pathway would be omitted. The set of plots available are defined from our Metapsych database, from the set of pathways and pathway classes available there, and also from the set of gene ontology terms. In particular, gene ontology terms are used more frequently when the dashboard is displaying transcriptomics data. Now, the dashboard itself allows the user to add or remove plots from any panel, and also there are many more metabolic panels when we're looking at gene expression data. What we saw right now was the dashboard, just now was the dashboard in Metabolomics mode, where it showed us only metabolism-related panels. And in general, you can click down on any plot to drill down for more information until you get to the level of individual metabolites or individual genes. So here are a few hints on using the dashboard. We suggest that you apply normalization and significance calculations to your data before you upload your data into the dashboard. And you can upload your data as a column delimited file in which there are metabolite names and or database identifiers in the first column, and you can specify using the import tool what other columns in your file contain data of interest. So essentially, you can have it ignore certain columns if you wish. And another thing you can consider is we found it useful to compare, to load into the dashboard all metabolites, and alternatively to load in only those that have significantly changed values according to the significance calculations, according to some threshold that you would like to define. So you can create different data files in which you've performed that thresholding, and only a subset of the metabolites are available in one of your import files. And one of the reasons we find it useful to make this comparison with the 2 different versions of the data is that sometimes the significance threshold will delete data that we believe is changing significantly, at least for data that we've looked at. And so you may want to be a bit looser because the dashboard allows you to assess changes yourself visually. And there are several types of data values that you can import. You can import fold change values or absolute quantities or p-values as well. And here's how you invoke the Omics dashboard from the Biopsych website. You go to the Biopsych homepage at biopsych.org, and then under the analysis menu, you can invoke Omics dashboard. And then you get quite a number of different options that let you choose to upload. In this case, upload a tab delimited file. You can select which file to upload, indicate that you have metabolite names in the first column, indicate which other data columns to use. You can enter something like 2, 3-5. You can select whether you have relative or absolute data and which sort of scale they use, and you can select among several different color schemes. Now, a little more information about the data set that we're looking at. This is a data set designed to get at the mechanism of action of a specific antibiotic, and this data comes to us courtesy of Dr. Paul O'Malley at SRI. And we're looking here at time series metabolomics investigation of the action of metronidazole against H. pylori compared to a DMSO controlled. And the overall study design is that 5 different biological replicates were taken at 5, 45, 180, 300, and 420 minutes after exposure to metronidazole began. And the metabolomics data processing, collection and processing was performed at Metabolome, who performed global metabolite analysis via LC-GCMS. They averaged the replicates and computed the fold changes relative to the control, and they computed significance p-values using two-way ANOVA with contrasts. And the thing to realize about this data set is that the first 2 time points occurred during the early phases of drug action when the cells were still viable, whereas the later phases of the later time points occurred when the cells were beginning to die. And one thing we could do through pathway tools, we see here what we call the cellular overview diagram that shows the full metabolic map of H. pylori as stored in biopsych. And overall, we were able to identify 318 metabolites in this data set, of which 103 of those were mapped to metabolites in the HPY psych database in biopsych, out of 602 total metabolites in that database. So that gave us a 17% coverage of the H. pylori metabolome. Now, another facility available within the dashboard is performing metabolite enrichment analysis, which asks which pathways have more perturbed metabolites than were expected by chance. And to use the enrichment analysis mode of the dashboard, the user has to provide a p-value column that's associated with each time point, as well as a threshold for what significance values to accept. And the user also specifies a multiple hypothesis correction function. And what the dashboard will do is to compute an enrichment score for every pathway using the Fisher exact hypergeometric test in this case. And the enrichment score that we calculate is the negative log 10 of that probability value. And what the panels that we're about to see will show are the enrichment scores for each pathway, as well as the highest component subsystem score for each pathway. And so here we're comparing on the left and right sides of this diagram the standard display of the dashboard that you saw earlier versus an enrichment mode display. And so on the left, we're looking at the metabolite abundance values, the fold changes, and on the right, we're looking at calculated enrichment scores, as well as the highest enrichment score present for each subsystem. So the enrichment scores are shown as the bars, and the highest scores present within amino acid synthesis. Here we're looking at the lines that are shown show us for all the pathways within, in this case, amino acid biosynthesis, what are the highest enrichment values for those individual pathways. So that's essentially telling us that there is more detail that's worth looking at inside this particular inside this plot. It's suggesting that we might want to click on that plot. Now, it's also worth noting that there are some differences between the left side and the right side. So for example, nucleotide degradation is showing very high enrichment scores compared to the fold changes shown over here. Now, in this display, we're going to look at the sulfur-containing amino acid biosynthetic pathways that are disrupted during the early phases of action. So what we've done is we started out at the biosynthesis panel, then we've clicked on amino acid biosynthesis to get to this display, and then we've clicked on cysteine biosynthesis to get here, and here we're seeing all the metabolites within the cysteine biosynthetic pathway and seeing that O-acetyl-L-serine seems to be undergoing the largest changes. And we can verify that here in the pathway display that shows us two different cysteine biosynthetic pathways and shows us that this metabolite is building up very significantly, suggesting that there may be a blockage right here. Now in this case, we're looking at a nucleotide degradation pathway. We've started by clicking on nucleotide degradation in the degradation panel, and we've then clicked on purine nucleotide degradation to get this display that shows all of the metabolites in this pathway, as well as plotting them here against the pathway diagrams. And so we're seeing that there appear to be two different enzymes that are blocked. In this case, even though there are 4 different reactions converting these 4 different metabolites into ribose 1-phosphate, they're all catalyzed by the same enzyme in H. pylori. And similarly, these 3 steps are all catalyzed by the same enzyme. So there appears to be some significant blockage going on here. In addition, if we look at the TCA cycle, we see that there's an accumulation of TCA cycle intermediates and of arginine as well in the later time points, which is consistent with as a signature for increasing morbidity. And so there are several new insights that have arisen from this dashboard analysis of the metabolomics mode of action study for metronidazole. In the early phases of metronidazole action, we see disruption of sulfur amino acid biosynthesis and of nucleotide degradation pathways. And it's also was known previously that H. pylori is critically dependent on nucleotide salvage, and the disruption of this choke point enzyme, nucleoside phosphorylase, is quite evident in the dashboard. And it's also known that genome stability is tightly linked to nucleotide metabolism in H. pylori, and this gives us a metabolic basis for the DNA breakage that was hypothesized to be occurring under metronidazole action. Now, that concludes the presentation of the dashboard. I'd also like to spend a few minutes talking about some other aspects of Pathway Tools. Let's start with a quick summary of the Pathway Tools software. Overall, this software allows us to create and maintain an organism database that integrates information about metabolic pathways with genome information, and for some organisms in biopsych with regulatory information. Pathway Tools has a number of computational inference tools, such as for inferring the metabolic pathways of an organism from its genome. It has interactive editing tools for modifying the database. It has query and visualization tools. It also has tools for generating quantitative metabolic flux models for both individual organisms and for organism communities. It has a number of omics data interpretation tools and a number of comparative analysis tools. And it's been licensed by actually more than 8,000 groups to date, and it's free to academics and licensed for a fee to commercial groups. And here's an overview of the capabilities within Pathway Tools. Overall, we can feed in an annotated genome as the central input to Pathway Tools, and its pathologic component will create a new pathway genome database that contains both the annotated genome and new information that Pathway Tools infers for that genome, such as the metabolic pathways of the organisms and operons for bacteria. That database can then be used in several different ways. The Pathway Genome Navigator can be used to query and visualize the contents of the database and to do different omics data analysis tools. The editor tools can be used to update the database, and the Metaflux component of Pathway Tools can be used to create a new pathway genome database, to create a metabolic model from the newly created pathway genome database. Here's the overall organization of a pathway genome database, which contains a different database object for each plasmid and each chromosome, for each gene and gene product, a different database object for each metabolic reaction and metabolite and metabolic pathway, as well as for the regulatory network of the organism. Okay, we've actually seen that slide before. I now want to show you the Cellular Overview tool within Pathway Tools, and in this case we're looking at the human Cellular Overview. The Cellular Overview is an organism-specific diagram that's generated for each organism in Biopsych, and it depicts the full metabolic network of an organism with biosynthetic pathways on the left, catabolic pathways on the right, energy metabolism pathways down the middle, and on the far right is a maze of individual reactions that have not been assigned to any pathway. Now this diagram is zoomable, so if we zoom in, we will see, if we zoom in through many levels, we'll get to this version that shows the name of each metabolite and enzyme and pathway. And in addition, we can import the same type of omics data files into this diagram as we can into the dashboard. We can import both transcriptomics, proteomics data, or metabolomics data. And in this case, we're looking at gene expression data. And if we zoom in now, we'll see a more detailed depiction of different pathways so that we can see more clearly which genes are having their expression levels changing. And in this case, we can, if we click on a given gene or protein, we will see a summary of the expression levels of that gene over time. So for this particular gene, its expression level went way down at time point 3, but then came back up again in time point 4. And for this particular metabolite, so here we're actually looking at metabolite and gene expression data together, because for this tool, unlike the dashboard currently, we can combine transcriptomics and metabolomics data into one data file. Essentially, with each line of the data file will contain data for either one gene or for one metabolite. So in summary, the Omics dashboard provides a top-down, organism-wide, system-level view of metabolomics data that lets the user quickly identify which cellular systems are changing in different ways, lets you drill down to examine the component subsystems in more detail. It's very well suited to visualization of enrichment analyses, and it complements other forms of metabolomics data analysis. And it's freely available at biopsych.org for the EcoPsych and MetaPsych databases, whereas a subscription is required for access to other organisms. And it's also available within the Pathway Tools software that you can download and install locally, such as to create your own pathway genome databases and apply these tools to them. And let me acknowledge my collaborators contributions. The dashboard was implemented by Suzanne Paley and Paul O'Malley, contributed the H. pylori dataset and analysis, and thanks to the NIH for funding this work.